Hi, this is Mr. Young, and in this podcast we talk about cellular respiration. And throughout our time, I want you to be thinking about two key questions. Those are, why really do you breathe, and why do you eat? The answer to that question is going to be within our discussion here on cellular respiration. As we start, one of the most significant parts of cellular respiration deals with this diagram that you see here. I want you to take a moment and see if you can respond to these questions. Number one, what is in fact this purple blob that's shown in the center? Using your knowledge from previous units, you should be able to identify this as, that's right, a membrane protein. This is the cell membrane, recognized by the phospholipid bilayer. And within, you see a structure that is allowing molecules, or in this case atoms, of hydrogen to go through. So this is a transport protein embedded within the membrane. According to this diagram, what is required to make ATP? Very good. At the bottom, ATP can only be made if we have its components ADP, or diphosphate, and a third phosphate, an inorganic phosphate, that we can combine to make ATP. Is this active or passive transport? Take a moment. And very good. You've determined passive transport because we're going from high concentration in the environment here to low concentration in the environment here. Passive transport. If you had to give a title to this diagram, what would you call it? This is obviously a diffusion of some molecule because it's going from high to low. Think about that question as we go throughout, and perhaps toward the end you might see how the title to this process relates to work we've done in the past with diffusing molecules. Lastly, if you had to name this purple blob, what would you call it? Remember, this is making ATP or synthesizing ATP. When you look at building or synthesizing ATP, we can use the term synth to help guide us. But then there's one more thing we might want to add to it. What do we call those molecules or proteins that help to speed up chemical reactions? That's right, enzymes. And what do all enzymes end with? ACE. And so this molecule we're going to find is ATP synthase. It builds ATP. So our question now is, how really do we do it? Specifically, how is this proton gradient formed that will then allow for the hydrogen ions to go through my ATP synthase to make ATP? Remember that question. How do we form the proton gradient? So in our time together today, we're going to talk specifically about glucose being the molecule for us to metabolize to generate ATP. We'll recognize that carbohydrates are not the only source of energy. Lipids, in fact, are the most dense and compact source of energy. There's more calories per gram of lipid than per gram of any other macromolecule. But carbohydrates are going to be the model that we will use as we look at cellular respiration using glucose. And so the key components of cellular respiration include using a glucose molecule, shown here, plus oxygen to generate ATP, water, and CO2. Now I want you to remember that when we are taking and really oxidizing this molecule to produce a 1-carbon compound from a 6-carbon compound, the components that we need in oxygen here may make us think about taking other fuels and burning them in the presence of oxygen to produce energy. Think about, for example, the production of heat that occurs when you take wood and you light it on fire. Wood, it's got carbohydrates inside of it. Think about all the cellulose and the other carbohydrates that make it up. In fact, when you light a match and in the presence of oxygen start to burn that wood, we recognize this is somewhat of a similar process. We need O2, we need some fuel to begin with, and then in the end we're going to create heat, and you bet we're going to release CO2 and water. We actually call that combustion. But that happens very quickly and in really one step. As opposed to the combustion process seen here with wood, if you take a look at my son devouring a piece of watermelon, we can see how the food will go into his body, and through respiration will lead to the generation of ATP as our source of energy. except as opposed to one massive step that releases a ton of heat that we saw in the burning of fuels like wood, here in a series of enzymatic reactions, with the help of oxygen being present, we're going to end up creating ATP. But notice how it's a regulated process with multiple steps to slowly break down this food molecule. So this is respiration. It may include a little bit of ATP as input, certainly enzymes, but along the way, it'll create the same products in the end, with the exception of ATP, as combustion. So when you look at all heterotrophs consuming things, whether it's this garter snake devouring a frog, as I heard one time when I was walking in the woods and heard a little as a frog was being munched up by a garter snake, or this gray jay sitting on my hand and devouring an apple after I called it in. In either of these cases, these heterotrophs, as they consume their food, have food products that are getting broken down in the digestive process. And the whole reason why we want to break them down is so that they can get in small enough parts to be able to then be absorbed from the food tube to the blood tubes eventually. That's what's shown in this diagram, where we're taking products from the digestive system, absorbing them into the circulatory system, and then from the circulatory system, assimilating those into body cells, where then the cells themselves can take molecules of food, or molecules derived from food, like glucose, and start to break them down. So right now, each of you should have this diagram in front of you. We're going to work our way through the process of cellular respiration by focusing on, first, the equation. Remember, to help us understand the equation, remember our two key questions. Why do you eat, and why do you breathe? Number one, why do you eat? Good, you eat to get energy, but we get energy from taking a food molecule like glucose and breaking it down into ATP. So that's our first response. We need energy, and we get it from the breakdown of glucose, in our case. The second question, again, was why do we breathe? And we breathe to get oxygen. As we breathe to get oxygen, we also need to breathe in order to eliminate waste products like CO2. But the last time you breathed out on a mirror or on a window, what else came out besides CO2 that you could identify? That's right, water. Water condensed. And so these are our key reactants in the process of respiration and our key products in the process of cellular respiration. The second thing we need to look at is trying to figure out how we can balance our chemical reaction. Notice up here, if we look at our reaction, we have 6 carbons on the reactant side, and yet on the product side, I have 1. Atoms are neither created nor destroyed. So whatever we start out with over here has to be over here. So now your job is, with this reaction written on the top of your paper, attempt to place numbers or coefficients in front of any of these molecules, reactants or products, in order to balance our overall equation. Give it a moment and try it out. All right, very well. You should have added a 6 in front of your O2, giving us a total of 12 oxygens, plus 6 more, or 18 on the reactant side. And on the product side, with a 6 in front of the water and a 6 in front of this carbon dioxide, you end up having likewise 18, or 6 here, and then 6 times 2, or 12, for a total of 18 on the product side. Balanced, and you'll check out your hydrogens are also balanced on both sides. So as we begin, we're going to trace the flow of food into the body. And this one step will summarize how food goes from your digestive tract, is absorbed into your bloodstream, and then is assimilated into your cells. And eventually in the cells, this glucose molecule will get transported across the membrane and is now found in the cytoplasm. Glucose, remember, has 6 carbons. So let's highlight right now food getting reduced to glucose, or excuse me, food getting broken down into glucose, and that glucose molecule then getting further broken down into a smaller molecule, a 3-carbon compound that we call pyruvate. But again, we got to conserve our carbons. So if we have a one pyruvate molecule that has 3 carbons, and we broke it down from a 6-carbon compound, there has to be 3 more. And so this 2 indicates we have 2 pyruvate molecules coming from 1 glucose. This is the process that we call glycolysis, and it will produce in the end 2 ATPs. So a quick summary of glycolysis that you want to record is the fact that glycolysis is anaerobic. An meaning without, and arrow is referring to the presence of oxygen. And thus, organisms that do not have exposure to oxygen can still produce ATP in the process of glycolysis. Likewise, you in your own body, when you lack oxygen supply, can still produce minimal amounts of ATP through anaerobic means, in this case, glycolysis. Note this is in all organisms, bacteria included, and finally the location is in the cytoplasm. Good? So what's next? We've got a pyruvate molecule. It's got 3 carbons, and we have 2 of them. It looks as though, remember, because there are energy in these bonds, that if we can take this molecule of pyruvate and break it down further, then we can get more energy out of it. So that's exactly what we're going to see happen here in a moment. A quick review of glycolysis, though. We're going to take glucose, break it down into pyruvate. Very good. You need to know that term. Again, how do we get ATP? You have to start with diphosphate and then tack on 2 inorganic phosphates to give you ATP. By the way, a quick note on the term glycolysis. Glyco is referring to sugar, and lysis is referring to the splitting up of the sugar. That kind of makes sense, doesn't it? Because here we're taking a sugar molecule of glucose and we're going to split it right down the middle. So now that we have pyruvate, we're going to be able to take that molecule and send it into the mitochondrion. That's what this picture shows. And it's going to go into what we call the Krebs cycle. The Krebs cycle goes beyond the outer membrane and the inner membrane and into what we call the matrix of the mitochondrion, the inside. And here in the matrix of the mitochondrion, we're going to end up taking our 3-carbon pyruvate molecule and breaking it down even further to a 1-carbon compound, that being CO2 or carbon dioxide. Now, carbon dioxide, as it develops and accumulates within our bodies after the cells have eliminated it, will get into our bloodstream, and our bloodstream will cause actually our pH to change. As CO2 gets in there, it will make our blood more acidic. And as our different chemoreceptors in the bloodstream detect that, we're going to be caused to breathe faster in an attempt to eliminate that blood, or eliminate the CO2 through breathing. So CO2 gets in our blood. We detect that. The blood goes to the lungs eventually, and the lungs allow for the gas exchange. In the process, though, what do we get out of it? We get ATP. And so a quick summary, Krebs cycle. It is aerobic, meaning oxygen is going to be required here, and it occurs in the matrix of the mitochondria. As you look at the Krebs cycle, there's a lot of nitty-gritty that we are not going to require you to know at this point. But I want to show you this diagram so that you can see the carbons involved, and that you can see also the key players of what we call electron carriers that are shown in blocks. If you look at what's really coming in to my Krebs cycle, you can see acetyl-CoA up here, a 2-carbon compound, getting assimilated with a 1, 2, 3, 4-carbon compound. And as that happens, we build a citrate molecule with 1, 2, 3, 4, 5, and 6 carbons. What are we going to do with those 6 carbons in the citrate molecule? Notice what starts to happen here as we break that down into eventually what you identify releasing CO2. And as we release CO2 in these 2 locations, we still maintain our 4-carbon compound, and this cycle continues and continues. But notice down here how we've produced ATP. In addition, we have produced some electron carriers because molecules came in that needed a hydrogen, and there they have a hydrogen. And we're going to pay close attention to the hydrogen stuck off of NADH and FADH2. So in the end, we fully digested glucose. We started with 6 carbons, and now we're left with 1 carbon. We can't break that down any further. But we only got a measly 4 ATP out of the Krebs cycle. What's the point of all that? Remember those electron carriers. We're going to get to those here very shortly and see why are they so significant. But as you look at those electron carriers here, as you follow a pyruvate molecule from glycolysis into the Krebs cycle, we see numerous electron carriers being formed that are going to play a role here later. Those electron carriers, NADH and FADH2. But what's the big deal? we're going to save them to be used in the electron transport chain. And the electron transport chain is going to have everything to do with what we started out with, trying to produce ATP from the flow of hydrogen across the membrane. So once again, if you look at all of the ATP produced, we started with pyruvate in the Krebs, and we were ending up with CO2. In that process, the 3-carbon compound was broken down into 1-carbon compound CO2. And to balance that, we have 3 of them. Yet in the process, we have electron carriers. There's 4 NADs and 1 FAD. End up picking up hydrogens. And those hydrogens end up serving as a form of an electron that's carried. Oink, oink. I want you to think of that electron as being deposited in a piggy bank when these electron carriers grab it. Oink, oink. We've got a piggy bank here for NADH and a piggy bank for FADH2. And what do you do with your piggy banks eventually? That's right, you bust it open and you spend it. In the process, we also made ATP. And we did this twice because remember we have 2 pyruvate molecules from 1 glucose molecule. Net gain then, 2 ATPs, and net gain 8 NADHs, 2 FADH2s. So the overall value of the Krebs, a net of only 2 ATP. Wow, this seems pretty weak. How can we use that to power us? Well, it's going to turn out that the overall value is going to be in the electron carriers. And we're going to use those here in a moment in the electron transport chain. Because we've deposited money in the bank, and we're going to chip them open here in a minute and spend it. We have to be able to do this, remember, because we need a lot of ATP. Specifically, we can burn up to 10 million ATPs per second by working our muscles. 2 ATPs from glycolysis, Krebs 2 ATP. Yikes, we are really struggling at this point. So there is a better way. And how do we do that? How do we make more ATP? This is it. It comes back to our molecule of ATP synthase, which is working on an established hydrogen ion gradient with a high concentration of hydrogens on one side of the membrane and a low side on the other. And then hydrogens flowing through this ATP synthase molecule, all to the end of combining diphosphate with an inorganic phosphate to give us ATP. So back to your diagram. Where can we show the electron carriers? That's our next step. We had our electron carriers here. We also had electron carriers from the Krebs cycle. Remember, electron carriers are NADH or FADH2. And the electrons are carried in the form of a hydrogen. Remember that hydrogens, after all, only have a single proton. And that proton is surrounded by, in a neutral molecule, an electron. Well, that electron is going to be significant to us, and that's what we want to focus on. These electron carriers go to the electron transport chain. And the electron transport chain is going to be a place for us to delve into now at this point. So those electron carriers we got from the Krebs cycle, we're going to take now those from glycolysis, which included NADH, and those from the Krebs cycle, which included not only NADH, but FADH2. Oink, oink. And now that we've got those piggy banks with the electrons in the form of the hydrogens, we're going to bust them open. So along to the electron transport chain we go. Notice where it's located in a membrane, specifically the inner membrane of the mitochondria. Not the outer, as you can see here, but the one right on the inside. And here at the inner membrane, we're going to see how we can take our electron carrier and we can use the hydrogen that was added and take that hydrogen with its component electron and proton and strip it of an electron. And as we strip it of an electron, all of a sudden we have leftover NAD+, and the electron is going to flow through and allow hydrogen ions to get pumped through. Did you see that one more time? The hydrogen that came from the NADH is going to go and cross the membrane. And as it crosses the membrane, it's doing so because of an electron that was carried by the hydrogen. The electron was chipped off here, leaving only H+ or a single proton, and that's what you have. This electron is going to move down this chain. It's going to get picked up by another molecule, This one is designating it Q. It's essentially like an enzyme of sorts, to transport molecule, and it's going to shuttle it from this site to the next site, a membrane protein. And as those electrons continue to be passed, hydrogen ions are pumped across. And finally, it's going to arrive at a destination at the end. So in each case, as electrons move, hydrogen ions get pumped across. They can't get pumped across on their own because, as we're going to find out, this is actually active transport. And we have to supply energy. It actually happens in the form of this moving electron now to pump it across. So not only does our electron carrier NADH serve in this role, FADH2 does the same. Hydrogen ions get kicked across, while the electron that came from the hydrogen is allowing the movement of the hydrogen across. So one of our questions we're going to look at now is just how the electron moves. Why does it move from one end to the other in the electron transport chain? From one, notice this, enzyme to another enzyme complex to another enzyme complex, cytochrome C oxidase, and in the end create water. So we built the proton gradient, but what powers the building of the proton gradient? What allowed us to build a gradient with a ton of H pluses over on this side and very few over here? That all came down to the name, the electron transport chain, electrons being the key component here. Electrons moved from one location to the next because they were pulled by an oxygen molecule. Why do you really breathe? You need oxygen to be the final wide receiver to receive the football of the electron that's going to move down through the electron transport chain and get picked up. Without oxygen, we do not take that electron through the electron transport chain. And as a result, if we don't take the electrons through, hydrogen ions don't get pumped across. And if hydrogen ions don't get pumped across, we don't have a gradient that will then allow for ATP synthase to work. So that is a huge deal. Why, in fact, do we get water in the end of our process? Because oxygen, as you can see here, is picking up our hydrogens. And in the process of combining, we make water. So electrons flow downhill to oxygen. So we did it. We established a hydrogen ion gradient. That's what you saw again right out here, where we were pumping hydrogen ions. We've got hydrogen ions that are here in the inner membrane space. And as these hydrogen ions are built up, we've created a gradient for now the flow of them downstream through our ATP synthase molecule. Once it's set up, they flow down, and it accommodates the synthesis of ATP as adenosine diphosphate is added to another inorganic phosphate to create ATP. Booyah! There we go. So our final thing, as we summarize electron transport chain, is to note oxygen's required. Write that down. As oxygen's put in, it receives the electrons as the final electron acceptor, and it leads to the formation of water. In the process, we're going to get a net of about 34 ATP. Wow! We had a measly 4 ATP from the other processes. Now 34. That's what it's all about. But don't forget, this happens because there's oxygen present. Without oxygen, this isn't happening. We're stuck with glycolysis. We're stuck with anaerobic respiration. Oxygen is the final electron acceptor. And lastly, the inner membrane is the location for the electron transport chain. And so on the back of this handout, you have these questions. Where did the glucose come from? The oxygen, the CO2. And where did they go? Where does the CO2 go? Water, ATP, and so on. I want you to look carefully at the equation and try to respond to these questions as a good summary of the process of respiration.